Atherosclerosis is the major underlying cause of heart attack and stroke. Despite the use of statins, cardiovascular disease continues to be the #1 killer. Hi, my name is Han-Jung Jo. I am Professor of Biomedical Engineering at Emory University and Georgia Tech and the Division of Cardiology at Emory University. I'll be discussing the role of flow on epigenetic DNA methylation in endothelial cells and its role in atherosclerosis. This work is led by my graduate student, Jesselyn Dunn, and my colleagues at both Georgia Tech and Emory University. The reason that we are interested in studying flow disturbance in atherosclerosis is shown in this slide. If you look where atherosclerosis occurs, it occurs typically in branched or curved arteries, such as carotid and coronary arteries. So we developed a mouse model called mouse partial carotid ligation model to test this hypothesis in 2009. This model involves ligation of 3 of the 4 small branches in the left common carotid artery. We left one branch open, superior thyroid artery. This surgery then induces flow disturbance in the left side. As you can see, there's a brief forward movement of flow followed by reversed flow. On the right side, however, flow is always unidirectional. Using this mouse model, then we tested whether flow disturbance can actually cause atherosclerosis. For this, we took ApoE knockout mice and we gave them high-fat diet. And within 2 weeks, you can see atherosclerosis developing in the entire length of the common carotid artery. And if you look at a time-dependent change in the cross-section, you can see 1, 2, 3, and 4 weeks, there is a severe atherosclerosis development, especially at 4 weeks. And we can also see intraplaque neovascularization and cholesterol cleft as signs of advanced lesions. With this model, we developed additional novel technique which allowed us to collect endothelial enriched genes directly from our animal models. Of the hundreds of genes, we noticed DNMT1. DNMT1 is DNA methyltransferase 1. This is an epigenetic master regulator. DNA methylation on promoter regions silences key genes. The role of DNMT in endothelial dysfunction and atherosclerosis, however, is unknown. We initially confirmed our DNA array data by immunostaining for DNMT1 protein. Next, we tested whether 5-ASA drug that inhibits DNMT can inhibit atherosclerosis. We first tested this in a partial ligation model where in control animals, they develop atherosclerosis, and by treating with 5-ASA in a dose-dependent manner, we can demonstrate that atherosclerosis development is prevented. In a different model, we also tested the effect of 5-ASA, in this case, a Western diet-only model without involving surgery for 2 months. They develop atherosclerosis in the aortic arch, especially in the innominate region, which can be inhibited by treating mice with 5-ASA. These data suggest that 5-ASA, by inhibiting DNMT, can significantly reduce atherosclerosis. To understand how 5-ASA inhibited atherosclerosis, we took 2 different approaches to understand the mechanisms. The first was microRNA to understand genome-wide mRNA expression patterns. As expected, flow disturbance induces a lot of gene expression changes, which can be reverted back toward normal state by treating animal with 5-ASA. In addition, we took another genome-wide approach to understand where this DNA methylation occurs on a genome scale. For this study, we used a method called RRBS, Reduced Representation Bisulfite Sequencing method, combined with next-generation sequencing. As you can see, flow disturbance induces hypermethylation in about 4% of the CG sites, which can be inhibited back to normal in about half of those by treating mice with 5-ASA. To understand which genes are regulated at the promoter region by DNA methylation dependent mechanisms. What we did was we combined these two data sets and used a systems biological approach to figure out which genes are DNA methylated on the promoter regions and which genes can be are also downregulated. By going through this exercise, we were able to figure out 11 flow-sensitive genes. And more surprisingly, 5 of those 11 genes actually contained cyclic AMP response elements. So these genes are HOXA5 and KLF3 and others. The importance of these cyclic AMP response element genes in endothelial dysfunction and atherosclerosis is currently unknown, but our initial studies do suggest that inhibition of HOXA5 can revert endothelial inflammation caused by flow disturbance, suggesting that HOXA5 genes and these cyclic AMP response element containing genes, as well as these other flow sensitive genes that we identified, could play an important role in atherosclerosis. So in conclusion, we showed that disturbed flow regulates epigenetics in endothelial cells by controlling DNA methylation in a DNMT1 dependent mechanism. This mechanosensitive epigenetic memory may lead to a long-term change in gene expression and atherosclerosis development. Our result actually could mean that when one exercises, especially aerobic exercise, this could change blood flow patterns, and this could impart long-term changes in gene expression by controlling epigenetic patterns. The role of epigenetics and the genes that we identified in this study could provide a new therapeutic targets to treat atherosclerosis.